Hello, my name is Elle Sitjar, and today I will be presenting my course project. What we'll go over today's topics: the introduction and the article that was used, the hypothesis, the data, graphs, confidence intervals, results of hypothesis tests, conclusion, and reference. The article that I chose went over the biomarkers of laboratory exposed northern leopard frogs to pesticides and what the influence was to their DNA cell cycle, fragmentation, and genome size. I wanted to go into why I chose this specific topic, so let's go to a little bit of background. I'm interested in this topic because I took a biology lab course last year and I thoroughly enjoyed learning about how DNA cells form. As I read through the research, I wanted to know how the exposure to the pesticides influenced their reproduction. My hypothesis. The alternative hypothesis is the mean length of testicular growth of the left testicle of injected frogs differs from 7.94 millimeters on the left testicle of the uninjected frogs. And the null hypothesis is the mean length of the testicular growth of the left testicle of injected frogs is the same as 7.94 millimeters on the left testicle of uninjected frogs. Here's the hypothesis using symbols. The first one using HA, sample 1 is not equal to sample 2, alternative hypothesis. And the second is the null hypothesis using symbols. The sample 1 is equal to sample 2. How the data was collected. The following data was collected on DNA parameters in whole blood, including cell cycle, DNA fragmentation, and genome size. It was also collected in a laboratory from northern leopard frogs exposed to the pesticides or insecticides. The data that I chose to compare was a sample size of 12 uninjected frogs, and I compared them to a sample size of 11 high-dose pesticides of injected frogs, or injected into the frogs. Here you'll see how I collected the data. On the left side here, you'll see that I compared them into, or I categorized them into groups, and then I compared the left testes length on the other side, on the right side. I have the uninjected frogs as my control group, as I can control them by not adding anything to them. And then the other group that I chose was the treat / high dose of the pesticide for the injections. On the right side of this presentation, you will see that they are almost overlapping. And then I will get into the results in just a moment. The confidence interval. So here you will see that the control, the uninjected frogs, the lower limit of the confidence interval is 6.64, and the upper limit is 9.23. I apologize. And for the treatment side, the high dose, the injected frogs, the lower limit is 6.92, and the upper limit is 8.69. So I am 95% confident that the average length of the left testicle of both groups of northern leopard frogs tested is between 6.64 millimeters and 9.23 millimeters. I performed a t-test, and at the significance value of 0.05, here you will see that the test statistic, or the z-score, is negative 0.18638, and the p-value is 0.8541. It is higher than the significance value. My conclusion is that the results are not statistically significant, and there is not enough evidence to reject the null or to accept the alternative hypothesis. It is possible that the mean length of the left testicle high-dose injected frogs is the same length as the left testicle of uninjected frogs. Here you will get my reference here. Thank you so much for listening.